Our next presenter is from 3P Innovation. He is Charles Pilato, and he is a sales engineer. Maintaining cell viability during cryovial filling is a critical challenge in cell therapy manufacturing. This presentation introduces cryoFIL, a cutting-edge automated cryovial filling platform designed to deliver high-precision, aseptic, and scalable solutions for cryovial filling. Welcome, Charles.
Thank you so much for the introduction, Perry. It's great to be back here. So if you can all fill out the polling question, that will be great. But I'll get into the presentation. So my name's Charles Pilato, and today I'm going to be presenting to you the cryoFIL. This is 3P Innovation's latest kind of technological development in the cell and gene industry. It's our cryovial filling platform. It's designed for GMP manufacturing, and it's for cell banking and reference sample applications. This unit, we've seen a huge, sorry, we're getting some comments. We've seen a huge amount of interest from this unit. You know, it was brought out about a year ago. And since then, you know, we sold units globally and we've seen a huge amount of interest. And we believe a lot of that has come from the developments in the cell and gene industry, in the cell and gene market, as more products are getting commercialized, as customers are moving towards more GMP applications, greater levels of sterility assurance. But we also understand that a lot of this is also come from, in essence, some of the discontinuation of the fillet, which is a current kind of lab scale automation piece of equipment that's out there. And as customers are kind of moving towards the GMP because that is currently getting phased out. And here today, what we're going to be focusing on is a bit around the design considerations of the cryovial filling platform. You know, when you're working in the cell and gene industry, there's additional considerations that you need to make when creating equipment and when doing that manufacturing process, especially around cell viability. So today we're going to be focusing a bit on the cell viability piece around the design of this equipment and how it's been kind of optimized for that. You know, a lot of what's done at the moment, it's manual filling. It's done by hand, by operators, you know, in biosafety cabinets, or it's done with lab scale automation bits of equipment. And what we're going to talk to you a bit about today is kind of the development we've made with the cryovial filling platform and how we see it as kind of the next step in the cryovial filling process, a step up towards commercial production, you know, from manual filling to automated filling and the benefits you see from an automated filling platform, and from, you know, from what is lab scale automation towards greater levels of sterility assurance, greater levels of in-process checks, greater levels of data traceability, you know, the whole GMP piece. And so we'll be talking to you a bit around all of those today. Oh, I've got to click next. So 3P Innovation. Who is 3P Innovation? So we're a custom automation, we're an automation provider located in the UK. We work with a whole range of global customers, and we do a huge amount of custom automation. And with that, we have our own product portfolio. And this focuses on specific needs within the market, for which the cryovial filling platform is the latest piece of equipment we've brought out, and which we're extremely excited to be talking to you about today. We do a huge amount of aseptic filling, if that's aseptic liquid filling and aseptic powder filling equipment. And it's that knowledge of aseptic processing, of aseptic manufacturing, which is what we're really trying to bring to the design of this equipment. You know, considering stuff like first air principles, considering stuff like material construction, making sure it's made out of materials like stainless steel, PEEK. It's got all the seals that you would expect from equipment of that nature, and also all the GMP and in-process checks that you would normally see. So the cryovial filling platform, in essence, it's an automated cryovial filling system. So we're uncapping, we're filling, and we're recapping cryovials. Now we're processing, we say up to 600 vials per hour. And the speed of this filling process is extremely important because, you know, when you're looking at the whole cell viability piece, so when you're doing the cell banking process, a lot of the time you're adding a cryopreservant like DMSO. Now DMSO, it's great. It allows you to do cell banking, allows you to freeze your cells. However, what it also does is when it's left just during the filling process, it actually kills cells, it degradates cells. So over time, you are damaging your cells, you are killing your cells. And so you normally customers identify what they call a critical window for which to do their filling in. And it's normally around the hour mark. So having that automation, having that speed allows you to fill more cryovials within that time. We've also got on the system, we've got a very high accuracy parasaltic pump. So we're achieving an accuracy of 1%. This pump has been specifically designed for the cryovial filling process. We've specified it for the filling of 5 ml fills down to 1 ml, which is the normal fill range we've seen within this market. And this filling, it's done into the cryovial, but on top of a high-speed weigh cell. So you will get 100% weight verification on your liquid fill. And all of that data, that's included within your batch report, within your 21 CFR Part 11 batch report. Now, the additional benefit of having your high-speed weigh cell for these weight measurements is that all that weight data, that feeds back to our integrated parasaltic pump, and that gives us the ability to do closed-loop filling. That gives us the ability to do zero-loss priming. So what that means is that at the start of the batch, when in normal filling applications, you've got to run a priming sequence, and that liquid, that's considered lost product, because you are, you're dosing it, and you're not going to get a filled cryovial. But within our system, we can do closed loop filling on that. So we make sure that your first vial, that is within specification. And same at the end of the batch, when you're getting the last of your product out, we can again do closed loop filling to make sure that those vials are within specification. So over the course of the whole batch, with a focus obviously on that initial priming sequence and the last of the liquid, we are making the most use and getting the most amount of specification vials within your batch. Especially when you're comparing it to systems which might have split tubing, where you're having to prime all of those different filling needles, all let's say 8, splits 8 ways. You've got to prime all 8 needles. You're wasting a huge amount of liquid. And in the cell and gene industry, where your product is extremely valuable, you don't want to be wasting all of that product. So this is the equipment as a whole. It's supplied integrated into a biosafety cabinet. And as I talked about earlier, because of the material construction and how we've designed it for aseptic manufacturing applications, this equipment, the automation equipment inside, can also be used within isolators for isolator applications. It is a rack-fed arrangement. Now, this is actually a really important piece of this equipment. We can use control rate freezer compatible racks. Now, what this means for your operators is that those racks of cryovials, when they're filled back into the racks, they can be taken straight from the unit, put straight into your control rate freezer. So you're not having to do any changeover of cryovials. You're not having to re-rack them from your original filled container into your control rate freezer compatible racks. So looking back at the viability in that critical window, you're not having to use kind of the end of that critical window, time in that critical window, re-racking. You can use the majority of that time and as much as possible of that time for filling your cryovials, because you can take those racks straight out of our system and put them straight into your control rate freezer. What's also, and you'll be able to see it, we've got a video coming up. So the rack here, it's a Thermo Fisher 161, but we can process a variety of different racks. This unit is designed to be very flexible, so we can process lots of different rack types and also lots of different cryovial types. We're using a peak puck arrangement, so they change that by hand, and they can be sterilized or they're autoclaved. So here you should be able to see a video of the equipment. Is that playing at all? Okay, yeah. Suppose it's playing. Yep. Here we go. I can see it now. It's very small. So what you'll see in this video is what we're doing is we're taking the cryovials from your control rate freezer compatible rack and we're transferring them over to the filling carousel. Here on the filling carousel, we're doing our uncap, our fill with 100% weight verification, and then our recap. Now, what's so beneficial about having those done at kind of sequential stations, so that you're uncapping a vial, you're filling it, and you're recapping it straight away, is the amount of time that that vial is open is as short as possible. We're not opening all the vials at once, filling all the vials and recapping them all at once. We are filling them one by one. And so it's another very important part to how we have kind of provided this equipment with the maximum level of sterility assurance, because the amount of time your vial is open is as short as possible. So a very quick kind of walk around the system. What you can see here, we've got our integrated peristaltic pump, and that's positioned next to our filling carousel with 100% in-process checks for that liquid fill. Here on the right-hand side, you can see our rotating rack. So that's a control rate freezer compatible rack. Now, this rotation means that we can pick from all the cryovials within that rack. We've also got our reject location. Now, this is really beneficial because what it means is that all of your cryovials, all the fill cryovials, they're in that rack. They are within specification, because any out of specification cryovials will be placed to one side. So you know that you can take that rack of cryovials, take that out and put that straight into your control rate freezer. And then obviously those vials, they're transferred over into your filling carousel. So what we talked about, what we're getting into here is a bit around the kind of the Annex 1 design of this equipment. You know, we've considered the Annex 1 regulations for aseptic manufacturing. So looking at the image here on the top left, that is a CFD analysis study. What that shows is the first air principles and how we're compliant with that. It shows how air is cleanly flowing down over your open vial. You're not having any turbulence. You've not got air coming in, picking up contaminants off other surfaces. It's clean airflow down. If you look at the image down on the bottom left, what that shows you is that hardware, what is actually above that open vial. And all it is the fitting needle and the filling arm. So it's very minimal, again, providing that really high level of sterility assurance, especially when paired with, as I talked about earlier, that minimal vial opening time. So looking at kind of the scale-up solutions, what we've shown here, this is a bit of equipment where it's been integrated into an isolator. This is actually a kind of 2-up solution, so twice the throughput. We also got the bulk feeding of cryovials. So it replicates the exact same process, really, that's seen on the biosafety cabinet solution, but just scaled up here within an isolator, which feeds into inline labeling and then re-racking via robots. And then finally, I'm just going to talk very briefly about a little case study, some work we're doing with our friends at Cellular Origins. And they're doing some extremely exciting work within the cell and gene industry. And what they're trying to do is eliminate all of the manual steps within the cell and gene manufacturing process. So they're creating what is a fully robotic suite of equipment, which is what they're calling the Constellation. And the cryovial filling platform is one of the bits of equipment that features within that Constellation, within those equipment there. And we've been working with them, kind of adapting the unit, customizing it for their application, basically to interact with their robots to do what your operators would normally do. And it's an ongoing bit of work we're extremely excited about, and hopefully there'll be even more to bring to you in the future. Thank you all so much for listening. I know this is the last presentation from the show, and yeah, please get in touch if you have any questions.
Thank you, Charles. Great presentation. So there are a lot of different vial manufacturers, solutions being provided to the industry. What types of vials can this system handle?
So in essence, we can process really any type of cryovial. What we're doing is we're holding it from below within our peak puck, and we're unscrewing with grippers from the top. So most cryovials out there in the market, they have what we would call anti-rotate features, but basically self-standing, or they've got grippers normally for the operators to hold on to. And we can grip onto those within our puck and then unscrew them from the top. And we use format change parts, just peak pucks, which allow us to process a variety of different types, all on the same platform.
How big is the system, cryoFIL? What are the dimensions of this?
Oh, I couldn't tell you off the top of my head, but it's small. So the image I showed you, I need to go back, but so it fits within a biosafety cabinet. We've actually can provide solutions within a 4-foot biosafety cabinet, but we've kind of settled on a 5-foot biosafety cabinet, which the equipment sits into, it's actually sunk into. And with a 5-foot system, you've got enough room to the side of it for your operators to maybe have your bag of product. Quite a lot of the time, they actually take it outside the biosafety cabinet. Having a 5-foot system gives you that space either side. 6-foot, you've got even more. But the system it's very small and compact, so it can actually fit within those small biosafety cabinets.
Yeah. And then are there wait times? Do you have systems ready to go if somebody's ready to buy one?
So at the moment we do have wait times. We're hoping in the future not to. As I said, this is a new product. We brought it out a year ago, and to be honest, we're just trying to service the orders we've got at the moment. But hopefully, as we develop forwards, we'll be able to reduce the wait times you would normally see on a bit of equipment like this.
Yeah. Yeah. Well, we hope this becomes much more common out there in the industry. And so the project that you highlighted with your current customer, that's an ongoing project.
Yes.
Any idea, like, what is the timeline for something like that?
Oh, sadly, I think you're talking to the wrong person. I mean, it's a huge project. It's absolutely massive. I mean, what we're doing here in the cryovial process is actually quite a small part about, automating the whole manufacturing process, kind of for cell and gene. So I anticipate this will be going on for a bit of time, and hopefully I'll be able to get back to you in the future and tell you about how this is all developing.
Well, great. Yeah, we'd love to hear about that. That'll be your case study at the next presentation in August, maybe. All right.
Definitely.
Well, thank you very much. Charles Pilato with 3P Innovation. Thank you for being with us today.